as a bacteria, it's administered as a freeze-dried preparation in an orally administered capsule. So it goes into the gut, and we know that it actually is able to stimulate the immune system in the gut itself, and that leads to downstream effects in the immune system, or systemically, and actually in the tumor itself. Now, in addition to the results that we've seen with the study that we reported on most recently, we've also evaluated MRx0518 as a monotherapy in a biomarker study where we've been able to examine how MRx0518 is able to affect the immune environment within the tumor. And what we've seen there is that we see an increase in tumor infiltrating lymphocytes, and we see an alteration in the ratio of the cytotoxic cells to the T regulatory cells. So we know that MRx0518 is actually able on its own to make that change to the tumor microenvironment. And what we've seen in the combination study, and the results we reported recently, we also talked a bit about some of the biomarker changes that we saw within that study, and it fits very much with the activity that we've seen with MRx0518 as a monotherapy. So, on treatment, we actually see an increase in T cells cytotoxic T cells, and a reduction in T regulatory cells in the periphery. So, all very much fits in with the mode of action, and that's really essentially the contribution that MRx0518 is making to that combination.